So it's my great pleasure to introduce Kohitij Kar, who is currently a research scientist at the McGovern Institute for Brain Research at MIT, working with Jim DiCarlo. Um, More importantly, he's going to be joining us as an assistant professor in July. And he's already been participating quite a bit in York activities. I've seen him at several seminars and talks, reading clubs. So we're at least I personally am very excited to have Ko join us. He does some very interesting work on neurophysiological investigations of visual intelligence in non-human primates and artificial intelligence systems. He does this very interesting blending of AI and neurophysiology, which I imagine he's going to tell us more about in his talk. And once again, I'd like to remind everyone that Ko is applying to be a member of the CVR. So faculty members, please send me your comments, I read your votes after the seminar. And with that, I would very much like to welcome Ko and take it away.
Thank you, Rob. Thanks everyone for joining in today. I'm really excited to share with you my research and my current research and my future plans. Also looking forward to joining York and coming over there and being part of you more closely directly in Canada. So today I will mostly talk about my current research that sort of leads into my future work. And it's about building like a mechanistic understanding of visual cognition. And so I would like to start by saying that, yes, I have thrown at you a few words like neurally mechanistic understanding visual cognition, and I will slowly unpack these and operationalize them as I go through my talk. I'll start by giving you a sense of the scientific questions that I'm interested in and what I think are the answers to this question. And I think I really liked sort of this way of sort of framing my research in my head also for futures, because I sort of start to think in terms of like, what kind of answer am I expecting? And then based on that expectation, I can devise like the methodological strategies of what kind of measurements do I need to make, things like that. So in light of that, I will explain to you my current research and my future directions. So some of you might have seen this before, but I'll go through this again, which is like, imagine, so my kind of like the sort of a naturalistic sort of viewpoint of like the main research strategy that we take in the lab. So if you imagine yourself walking through Fifth Avenue in Manhattan, so as you are walking through, you parse the scene by making small eye movements, fixating at different parts. You can fixate here and you can say like there are two people coming towards you. They're probably like friends because they're very close to each other. Here's another person on their phone, looking around, maybe looking for some direction. Here's another person on their phone, but this time you might infer that this person might be texting or on social media. Something. Here's another person on their phone. So you start to believe maybe that there's too much people like just walking, looking at their phone. You walk ahead a little bit. You can tell time very quickly. You can look at this clock and you can say, okay, it's something 10:40 or so. Specific images might sort of bring emotional reactions in you. You can find this tempting. It can also trigger memory. Like you can remember what happened when you had this last time. So you decide to like sort of move on and keep walking. So as you keep walking, you have a sense of your own direction, and you have also sense of direction of other objects in the field. And so based on all of these sort of visual factors, you can sort of reason and you can say like, okay, I can see like there's 12 seconds left to maybe cross the road, but this is Manhattan. So given an opportunity, the car might try and hit me. So you stop, so you make a decision. So like, this is what to me is like visual cognition in play. So all of these things together is what I think humans are capable of, very sophisticated visual cognition. And so this is something I'm super interested in exploring, but obviously in the lab, we cannot do experiments with this kind of stimuli to begin with. So we kind of like parse this into independent components and try to tackle it separately. So the first component that I will talk about today is core object recognition. This is the capability of the system to sort of like identify the objects that was presented on the retina, on the fovea very quickly. So I can quickly show them and you can still recognize them. The other parts of this of the task could be like recognizing the movement of the objects. So the people moved in this direction, the cars moved in the other direction, or sort of the complex reasoning that I was telling you about, like basically figuring out like what to do based on input that is provided from the visual sensors. So today, most of my talk will sort of rely on my current research, or talk about my current research of core object recognition. And as a future direction, I will mention at the end, like how I want to sort of take the same approach towards more complex visual tasks, higher level visual tasks. And so the main question that I'm asking is, how does the brain solve such visual cognitive tasks? So that's the question. So what is the answer to this question? So I will bind everything, my entire talk, in the framework of core object recognition. So this is the overall behavior that we have operationalized in terms of specific tasks, but everything that I will say will be sort of bounded by this kind of framework of core object recognition behavior. In the lab, I can do a few things. So I can get behavioral measurements. I can make behavioral measurements in humans from crowdsourced ways or in lab behavior. I can make behavioral measurements in monkeys. So we have this system in the lab where this is something I'm also going to have in my own lab, where monkeys will be doing the tasks in their home cages on tablets, very similar to humans, how they do in their home environments. Because I'm a neuroscientist, an experimental neuroscientist, I can also look inside the brain. So I can, based on literature, I can start probing different parts of the brain where these tasks might be implicated. And I can do a couple of things. I can record. I can also perturb activity from this on these areas. And so I can get a lot of data this way, both behaviorally and neurally. But at the end of the day, things like progress, knowledge, mechanism, understanding, and the answers that I was telling you about, they will all boil down to like, what kind of model do I have of the system that I'm trying to study? Models that explain and predict both the behavior and the neural activity. And I can only assume that these models are going to become more and more and more complex as we start to like get into more fine-grained behavior and like more fine-grained details of the brain that is implicated in this behavior. So these models can start off by being models like HMAX, some of you might be familiar with, or SIFT, or even like word models. Like when nothing is available, we can just rely on words like brain area X does face processing or something like that. They're also implicitly models. But then by the end of 2014, when I joined the lab, the kind of models based on like different behavioral studies and different neural studies, where this class of models called the feed-forward deep convolutional neural network, specifically for this kind of object recognition task, they were, at least in some circles, they were the state-of-the-art model for explaining the behavioral and the neural data. So I would like to first give you a little bit of insight of like why was there all this hype around specifically these kind of models. So the first thing about this kind of model is that it gets the job done. So if you show an image of a dog, for example, here to this model, the model takes in this image, processes it, and spits out an answer, dog. And it does it not only for this dog, but for this dog, all of these other dogs. And it might be done very seamlessly for humans, like we don't even think about this kind of an issue, but it used to be a computationally very challenging problem to solve. And these type of models started to really solve this problem at a very large scale. So that was the first thing, that at least behaviorally, they were very high performing. And then, of course, if you buy into this sort of hierarchical structure of the brain, specifically the visual brain, going from V1 to V2 to V4 to IT, you can find a similarity between the hierarchy of the brain to the hierarchy of the model. And some of the most seminal work sort of done also in Jim's lab with Dan Yamin's kind of was first to show sort of this like correspondence between regions of the brain and the hierarchical regions in the ventral stream and the hierarchy of these models. And actually, currently, there are also studies from other labs showing the same thing in V1 and V2. Someone's microphone might be on, making some noise. To make sure. I hope it's not mine, because I feel like a kind of rustling kind of sound. Yeah, so there is this sort of correspondence between these two systems that makes these kind of models specifically appealing for someone like me to use as starting points of experimental design and probing the system more efficiently. Okay, so this talk, the first part of the talk about my current research, I have sort of divided into 3 sections based to just keep track of like what I'm talking about. I think I'll try to sort of package the talk into 3 questions, 3 approaches. So like, how good are the current models of vision? What are the necessary missing components that we have identified? And like, what additional types of experiment can we do to provide more insights and improve the current models? So these are the kind of like very broad topics that I have hit upon during my postdoctoral work in Jim's lab. And this is what I want to mostly present today as part of my current research. So let's start with the first question. How good are the current models of vision? So if you look at this model that takes images, and like there's V1, V2, V4, and IT maybe in the model, some correspondence to it. And if you look at any neuroanatomy 101 picture of a brain, this model is sort of like a joke, because it's like it has like so many components. The brain has so many different components that these models have no mapping to. So it is clear that this model is not the brain. It's not anything like what the brain potentially is going to look like in terms of a model. So it's incomplete, obviously. And so this quote of George Box is very relevant here. Is like essentially, if obviously all models are wrong, but some might be useful. So one of the approaches for us was to sort of see where, in what domains this kind of models can be useful, and what can it actually bring us in terms of understanding the system or probing the system in a different way. So in one of my studies, I was recording in area V4 with this kind of Utah array electrodes that I will also show you more of this kind of data later on. But like, we were recording in V4, and as I told you before, we can map parts of the brain to parts of the model. So you can have a model that starts to predict very specific neurons that you're recording on a given day or on a given month, for example, also. So one thing you can do with this model is you can make held out predictions from this model. So the first thing we do is predictions. So you can, I'm showing you a plot where the x-axis is the predicted firing rate from the model, and the y-axis is the measured firing rate that I actually measured in the brain. So as I'm showing you here, that they kind of like align with the unity line. So like the model is making predictions that are actually sort of holding up in the measurements that I make. So that's the first kind of utility of this model, to like make predictions that can be verified, validated, which is also like a test of the model. The other thing that we can do is control. And this is something that I think having this kind of image computable explicit models really allow us to do beyond intuitive sort of like, understanding of the system. So what do I mean by control? Is essentially the question, what drives neurons in area X? So this is one form of the control question. And I want to tell you that this is a very common question in systems neuroscience, in visual neuroscience. Like, people have asked this question before, like what drives neurons in V1? What kind of images drive V1 neurons in V2? What kind of images drive neurons in V4? So on and so forth. So the idea is that we want to make predictions of like what kind of images to show that takes the activity to sort of this levels, like much higher levels. And as I was saying, that there are known stimuli patterns or patterns of luminance that are known to excite different areas more than other areas. So like oriented Gabor's for V1, textures for V2, complex curvatures for V4, and sort of faces we know are some stimuli that drives IT responses. So I was in V4, so there is a sort of challenge set, for example, for us, like if we claim that these models can tell us more than what these stimulus can sort of like excite the neurons, then there's something to be maybe gained there. So what we did basically is that we first showed these specific images to different neurons. Here I'm showing you for this particular neuron. There are some images that made the neuron fire kind of higher, although there's a side point that there are some other naturalistic images that might have already made the neuron fire high enough. They might have components of this kind of curvature in them. But what we wanted to test is that having knowledge of the neuron and of the model, like, can I synthesize images that actually push the neurons at higher levels? So the idea is to go back to the image space and try to manipulate the pixels in a way that predicts that the V4 neuron that I'm measuring from in the brain will be activated at much higher levels. So this is what we did. So here's a couple of examples for this neuron. And we end up finding that we did achieve like unprecedented levels of like high firing rate and this idea that we will find this kind of super stimuli for this neuron. Now we can look at these stimuli all day, and we can make up theories and stories about like what they look like, what are the kind of words associated with this stimuli. But ideally, what we want to make sure is that like these stimuli are basically these red dots. They should be on the unity line. They should not fall off the unity line. So whatever the model predicts, that should happen. So that's kind of, we consider this as a failure of the model to some degree to actually predict it very well. But nevertheless, these images drove the neurons way beyond whatever was known before to be driving this sort of system. So I'm just telling you that there's a way that the field has perceived this work of like, oh, these images are cool, but like for us, it's kind of like a failure, and also like it's a proof of principle that you can do stuff with the model, but it's kind of a failure of the model because like the model is not able to really point us to the specific part where the neurons are gonna like land on. It's this kind of technique is not only used for one neuron. We did this for multiple neurons. So we had a population of neurons. So we could set the goal as like, let's target neuron, I would say like, let's say your target neural site 12. So the goal would be like, find a stimuli that sets neuron 12 at very high level, puts everything at very low level. So that could be another kind of control goal. And we had like reasonably good success in like generating stimuli that does this kind of stuff. But again, as you see, like there are these other neurons here that sort of started to fire more, and one might start to speculate why that is the case, because maybe neurons are connected in ways that make them sort of dependent on each other. These are not things that are present in the models. So there are many ways in which to improve the model, but currently this is one application that the model can be used for. So like, imagine this neuron was a neuron that drove like emotional behavior, and you're targeting that specific neuron with parts of the pixel changes that could be useful in a clinical setting. So taking this work a little bit forward, we have started exploring also what kind of stimuli might excite IT neurons, for example. They look fundamentally very different from V4 neurons, but again, this is work in progress. So this is sort of like something that I would also pursue when I start the lab, and we have some recordings in the IT areas. Again, this is a kind of like work that I think could potentially lead to clinical translational, has a clinical potential in it. So are we done? Obviously no, because I've already showed you there are many ways in which the model is failing. So I would like to kind of say that prediction, there was like 50% variance explained from other papers, and also what we have shown, and control also good, but not perfect. So it's like a glass half full kind of situation that we have in our hand. So that kind of brings me to my next question, is like, okay, what kind of components are missing currently in the model that we could have a good grasp on, and like we could, incorporate in the model, and the model can become better predictors and better controllers of the system. So I kind of list them out a little bit here, like, so known components of the brain that are missing, and I'm sure all of you can add more components to this. So for example, these models are completely feed forward. They have no dynamics in them. There are other visual brain areas. It's not just V1, V4, IT, and V2. Recurrent connections are not there in the model. The brain is heavily recurrently connected, we know. IT and other areas have a very specific topographical organization that these models currently don't have. Learning mechanisms of the model are quite non-biological, and that's something that everybody's aware of, but like, because these models work so well, we don't really focus on that as much. Dendritic computations that happen in the brain doesn't happen. There's neural noise correlation and many other factors. So there are a lot of things that these models don't kind of like align with the brain on. So for me, when I was approaching this question, I sort of like picked my favorite ones that I thought experimentally I would have kind of a good handle on like contributing to. So like, I took this 3, for example. Some other folks in our lab has taken up like topographical organization and learning mechanisms as part of their goal. But like, I took kind of like dynamics and recurrent connections and the role of other brain areas as sort of like the things that I wanted to contribute to. So what was the approach? The approach, again, just to reiterate, for me, was like, I have 2 systems, and I want to see like, what can I bring from the system in the top to the system in the bottom to make them align in some goal. And the goal to start off was just behavior. Like, so the idea was like, let's take this system, which has just the feed forward component of it. And we know that there's feedback connections in the brain that doesn't, which is not part of these models. So can we find any evidence that these feedback connections and the recurrent connections are involved in this particular behavior, core object recognition behavior? So what we did first is to answer this question, that are the recurrent circuits functionally relevant for core object recognition behavior, we compared the behavior of these two systems, just like made the two systems, monkeys and models, and also humans in this loop, perform the exact same behavior. And I will tell you what the behavior is, and compare their behavior. So the behavior was very simple. So we have a human or monkey fixating at the center of the screen. An image will show up with an object, and then there will be two choices. Like, so if you had looked at the image, it was an image of a face, so you have to select face in this case. And another example would be of a bird. So if you have paid attention, it's an image of a bird. So in this way, we can generate like multiple tasks based on different objects, and also multiple thousands of images of those specific object categories. So I had used these two strategies to develop the image set. One was the synthetic naturalistic images, where we had full control of where we are placing the object, what orientation, what size. And the other was more naturalistic settings with photographs just downloaded from Microsoft Coco database in this case. So here's an example of like comparing these two systems. So on the x-axis is AlexNet, which was at some point the leading model in the computer vision field. And on the y-axis is monkey performance. So I'm comparing performance of monkeys with performance of AlexNet, and each dot here is an image. So it's like you should think of the axis as like, how difficult is it to tell the object present in that image? So that's kind of what we are trying to quantify in this. And so just for clarification, just for like simplicity, I will try to like divide this into two image groups. One is the control images, where because this lies on the unity line, it's like AlexNet is bad, monkeys are bad. AlexNet is good, monkeys are good. So it's like model follows the monkey or monkey follows the model. That's the control images. The challenge images are the ones where the monkeys are much better than the model. So the idea is that there's something going on in the monkey head, or multiple things going on in the monkey head, that the model has no access to that machinery, so it's failing to do that. So within here could be like a subspace of images where the monkeys are better because they have recurrent circuitry available to them. So this is what we wanted to test, whether we can gain some evidence towards it by doing neural recordings and looking at what is actually happening inside the monkey's head in this kind of setup. So what I did was I implanted electrodes in IT and V4 across different hemispheres of multiple monkeys, and we started recording these signals in their inferior temporal cortex, where the objects are potentially solved, and while the monkey was doing this behavior. So as I showed you before, this is the Utah arrays that we implanted in these areas, and we were recording the monkey brain activity. Just for sort of simplicity, again, like I would like to focus only on like a small band of behavior. So let me go through, let me pick an image from this set and go through a little bit of like how we looked at the data, how we looked at the neural data as we were analyzing and trying to figure out what's going on in the brain. So the way I looked at the data is like, imagine you're recording from all three. These are the arrays that are placed, like each array has like 100 contact points in the brain that it can record neural activity from. So I show this image to the monkey, and I'm recording from the array. And so the neural, let's say the neural activity is sort of evolving over time. Usually the way we have looked at the neural activity is very common, is dividing, coming up with this vector called the population vector, which is basically like counting the number of spikes in any given time bin. So in this case, I'm showing you a 10 millisecond time bin. And again, here's the main analysis, and I think it will become clearer as I show you more of this. Is essentially like we take the population vector and pass it through like a decoder that tells us like how accurately can you, given that population activity, decode what object was present in this in the image. So if I pass through the entire dynamics of the system, you will see at some point IT neuron starts to become active, and this IT decoding accuracy also starts to rise up, and it hits a point. So what is this point? This is basically an estimate of like just empirically estimated monkey's accuracy for that specific image. So this is how accurate the monkey was to determine like what object was there in that image. And so what I'm doing here is that sort of getting an internal read of like how IT was able to solve that question compared to like how it was solved later on by the monkey. So this kind of in a way is like reading the mind of the monkey, because the task is done like sort of way afterwards, like around 500 milliseconds or so, the monkey is actually providing the response. So between 100 to 200 milliseconds is when the monkey has just seen the image, and I'm trying to just sort of like figure out what is happening internally. So this is for one image, and the time that is shown here is we started calling it the object solution time. It is like the time at which the solution in IT is similar to what would happen in behavior. And we can estimate that for multiple images. So we can pick another image, you can have the same solution, same analysis done, you can find a solution time. What was interesting to us essentially was that if you start picking neural images from the challenge set, here's one example, here's the other example, we started to see that although the neurons started to fire almost exactly at the same time, the solutions, that is the linear formatting of the population vector that gave rise to the object solutions, they were developing slightly, they took like slightly longer to develop. And this was to us like at least putative sort of an evidence that there must be something done to the formatting of the, there must be some additional requirement, additional transformations required to the population vector before it becomes sort of linearly separable in terms of object identification. And this was our first sort of hint that maybe these images, some of these images, maybe not all of them, some of these images are taking longer because this reformatting needs to happen, and maybe recurrence is a mechanism through which that reformatting is actually happening. So this data cannot completely identify recurrence as a mechanism, but it points towards that direction. So this is at least consistent with the idea that additional time might be, equivalent to like some form of recurrent computation. And this is what I'm showing here, just to reiterate again that the firing rates for both of these images, the latencies were very similar. It's something happening at the later time bins, which were mostly things that were different. We did this for multiple images, and on average, there was around a 30 millisecond delay between the challenge and the control images. And here's a histogram of the two distributions of images. So, and based on different other controls, we infer that this additional compute time might be indeed due to some recurrent processing that is critical to identify such challenge images. So in fact, beyond the two groups of images, we could also make like a general statement that recurrence might be more critical for images which has longer solution times. So that could be another way of looking at the same data if you don't want to specifically rely on this grouping and just try to look at individual images themselves. So this idea that additional compute time equals recurrent processing takes a little bit more weight, I think, after this experiment. And also because now we have this independent measurements of each of these images and how long they take to be solved in the brain. Imagine I'm making a recurrent neural network now, they become sort of the target of like what is expected out of that recurrent neural network. Okay, so I think to some degree, maybe I have shown that dynamics in the system and recurrent computations in the system are critical for this specific core object recognition behavior. Now, obviously, this is a no-brainer that the brain is dynamic and the brain has recurrent connections. So like those are important for behavior. Like I think I cannot argue for that. The reason why we wanted to do these tests and to sort of make sure that they're important for this particular behavior is again because of the framework that the way we want to sort of progress is like, okay, solve one task, then make it bigger, then go to the next step. So like, is dynamics and recurrent an important component to solve it? And here I'm showing that maybe they are. So just to kind of go beyond this a little bit, because I'm an experimental neuroscientist and I'm interested in understanding like which part of the brains are really implicated in this sort of recurrent computation, I went a little bit one step beyond and asked like which recurrent circuit motifs might be relevant. So if I kind of draw out the circuit diagram a little bit more elaborately of the ventral stream. The ventral stream has a lot of different connections to like downstream areas, different connections within itself, bypass connections. So there are multiple sources of this kind of additional compute time to sort of seek into the system and to be available at IT. So the way to tackle this, or one way to tackle this that I thought kind of was feasible for me, was that I could pick one area, and I picked PFC to IT or PFC to ventral stream recurrence, and I can start asking like, is this particular pathway, is it relevant to like developing those kind of late stage responses that I was witnessing in IT? So that's kind of like one of the strategies that I took. And why PFC? Because there has also been a lot of studies done in ventrolateral PFC showing visual responses in those areas. And this is also something related to my future interest in like going into more high level cognition. So I thought this would be a good area to sort of like start looking into in general. So I did an experiment, and very simply put, the experiment has it's not a very complicated experiment. It's just the whole idea is to ask like, are top-down feedback from ventrolateral PFC critical to this behavior? And the idea is that, okay, I'm showing an image. The very simplistic model is that the image hits the retina, goes to like thalamus to V1, goes to the ventral stream. There are connections to the ventrolateral PFC. So maybe it receives input from ventrolateral PFC, it receives input from IT, and then there's some kind of feedback that goes back into the system. This is like a very simplistic, diagram of the system. So what I did was I perturbed the system. And the whole idea was that once I perturb ventrolateral PFC, and I put mucimol in this case in ventrolateral PFC, maybe this feedback that goes from ventrolateral PFC to IT or the ventral stream will be weakened in some ways. And if that weakening has any impact on A, the IT representations, and B, the behavior, then I can start to make the point that maybe ventrolateral PFC is actually a key component of this recurrent circuit. So the idea was to actually repeat the exact same experiment with IT arrays with Utah arrays in IT and the monkey doing the behavior, but now with two conditions. One is like nothing exactly the same as before. The second one was while I injected mucimol in the ventrolateral PFC area. So what did I find? This is a sort of a brief diagram of the setup. And just to recap the hypothesis was that maybe if I do this experiment, because I kind of ended up with this hypothesis that recurrence might be more relevant for images with longer solution time, it might be the case that if I perturb VLPFC, it would be the late solved images, the images that take a little bit longer to be solved. They might be the ones that might show more deficits. And if that's the case, then that is one way of linking the recurrence story to this perturbation experiment with VLPFC. And this is kind of similar to what we found. We found this sort of differential effects between early and late solved images, both in the neural activity in IT, where late solved images showed more deficits than early solved images, and in behavior, where we observed exactly the same idea. I'm going through this quite fast because I'm kind of avoiding a lot of details of the analysis, but there's a couple of key points here. One is that this is not recording from PFC, right? This is recording from IT. So one could expect no change at all in IT when you're perturbing PFC. So the fact that there is a change, and there's a change that is similar and kind of like predicted by the recurrence hypothesis, to some extent is very much confirmatory of the fact that maybe this circuit is involved in a recurrent way for this particular behavior. And the other kind of meta point is that there are some overall behavioral deficits that we see when we inject mucimol in PFC. So this kind of data might also start to give us more insight into like, how is the behavior executed overall? Like, is it a read from both PFC and IT, or like just PFC? So that could be these kind of research question that is also coming out of some of this data. So again, I just wanted to briefly mention that the main take home is like, yes, we kind of found some data that is consistent with the idea that VLPFC and the ventral stream might be bidirectionally communicating to execute core object recognition in the lab setup that we have currently. So if I go back to just the experiment and results that I have done so far and go back to the initial framework that I proposed. So what's happening with respect to that? So how does the brain solve core object recognition, right? Like, so I showed you, I did some behavioral experiment. I got some data from that behavioral experiment. I did some neural experiment, both recording and also perturbation again. So I got some data from there as well. So what happened to the answers? So I'm trying to show like this might be my way of like improving or progressing, making progress in the field, which is like what happened to the answers. So we started 2014, as I said, with AlexNet or some kind of feed-forward network. The first experiments sort of started giving hints that, well, the system has to be recurrent to really fully capture the behavioral data and the neural data. And then over time, I also added this other component to this model, which is like the ventral PFC node. So these are architectural insights to improve the model, but I hope that the data that I've collected and the grain at which it was collected will be used to do two things. One, it can be used to directly train the model. In some sense, this is something that we are trying right now. The other is it can be used to guardrail the model to show that like you can build a recurrent model in many ways, but not all models will lead to the exact same sort of ordering of the object solution time across those 1,000 images. So those can be, so this data can be used to really put the models, put the new recurrent models to stronger tests. And this is exactly what we are doing. I'm doing in collaboration with two groups, one in Jim's lab with Martin Schimpf and Jonas Kibulius, and also with Dan Yamin's group at Stanford with Aran Naibi and Dan Baer. So like we have a newer class of models that are recurrent in nature, and we're trying to see how those are faring with our older data. And we are in the talks to how to put in the VLPFC module into this system. And this is something I'm also going to continue working when I join York. I'm already collecting neural responses in VLPFC, similar to what we have done in IT. And I hope that that kind of data is also another way to constrain the models, because that data and how it relates to IT activity over time would be super, I think, interesting to look at with respect to this entire problem. So that's kind of like some of the sort of pilot data that I have, and I will be working very closely with when I start my lab at York. So another small technique detail that I've worked on, and I think is going to be very useful going forward, are some of these chemogenetic techniques that I've developed in the lab. And so before I talk about the technique, you could ask, like, why do we need another technique to answer the same kind of question that I was already kind of telling you all about? So let me motivate a little bit, like why newer techniques are actually important and necessary to develop. So in the previous experiment that I showed you, the type of connection that I was trying to perturb were connections from area A to area B, which is like in that case, it was PFC to IT. It was these projections that go from PFC to IT. These are the kind of projections that I was really interested in perturbing. But PFC doesn't really project only to IT, it projects everywhere else in the brain as well. So when I was injecting mucimol, I was basically affecting all of these connections. So this was not a very targeted experiment. We really want to just target this particular projection pathway. So in the last 5 years, on the side, we have been trying to develop some of these techniques in macaques where we potentially can have a way of targeting very specific projections in the brain. So the core of the idea is basically we inject a particular virus, a Cre virus, on this end, on area B, that is taken up by the axons of area B up to area A. And then we affect area A with another virus that is Cre dependent and has some other capabilities of silencing the area or activating the area, so that when these viruses injected, it will only target neurons that have transported the Cre from the other area. So that way, when we infect these neurons, it will only have effects on this particular projection pathway. That's kind of the overall idea, and it has been worked out quite well right now in rodents across many studies. It's just in macaques, these kind of techniques are quite underdeveloped. And so what we have done is like kind of approach it in parts, like try and see what the Cre viruses are that work well. This is a paper that we published. Like this is like has information of many viruses. Some of them have worked, some of them haven't worked. I'm also developing this kind of Cre dependent DREADS viruses, but also just a DREAD virus any ways to kind of like perturb the system. Again, dreads is a concept that I haven't spoken to you all about. But again, the whole idea is to inject something that can manipulate the neuron, either increasing its excitability or reducing its excitability. That's the global idea, and these are sort of techniques to do that. And if you're interested to hear more about this, I gave a talk at VSS this year, and the video is available. So I've gone into a little bit more details of the kind of techniques that I've used to sort of probe the system. So I don't want to go into the details of it because it's completely out of scope for this talk. But if you're interested, I'll be happy to talk more about this as well. All right, the last part of my talk, future directions. And I was very excited to build a lab website for myself. So if you haven't seen it, please go and check it out. It's called Vital Colab, Vital standing for Visual Intelligence and Technological Advances Lab. And Colab is co and lab, so kind of also collaboration. So I think the spirit of collaboration, this name, I kind of like it a lot. And I have never been to York, as I said in the beginning. And so I'm assuming that when I reach there, I'll see something like these pictures. And so I'll be super excited to get in there. As I have already demonstrated in this talk, there are multiple techniques that I want to span in the lab. And they're very interrelated to some degree. And the only technique that I haven't really spoken to you about here is this idea of clinical translation, and the fact that the clinical translation and the neuro AI crosstalk kind of works together. And I'm not, unfortunately, going to talk about it much in the later slides. I've spoken to you individually a little bit about this, and I would be happy to discuss more about these directions. But this is something that I'm not going to talk about in the next few slides. But hopefully I've given you an idea of like the primary electrophysiology work, the NHP behavioral work, the human behavioral work, and the in vivo sort of genetic approaches that I'm taking to sort of answer some of the questions. But maybe what I want to spend more time in the last few minutes here is like, what are the questions? What are the kind of questions that I will try to answer? And what are the motivating factors for asking the questions in the way that I'm going to ask the questions? So maybe you've seen this video before, but I would like to show you again. It's a really, I grew up in Kolkata, where there are a lot of monkeys, and in the temples and other areas, monkeys are always running around. They do all kinds of mischievous stuff. And now that I'm working with monkeys, I kind of understand why my mom used to call me a monkey when I did some stuff. So like, monkeys are, very smart creatures. And I have to say this, I was super disappointed when I joined Monkey Lab at US, and I saw that the tasks that the monkeys were being asked to do are like this dot motion discrimination, or like, grating pattern orientation discrimination. Like, this is such a very diverse and rich species. Like, why are we not really utilizing it? But then after 5 years of work, I now realize why we are doing that. So I have no complaints about that right now. But still, there is some kind of inner wish in me that I want to, when I have my lab, I will like to push these domains a little bit beyond some kind of simplistic behavioral domain. So this video might kind of motivate why I think that. So this is a setup video where the guy is completely setting up the monkey. This is in some temple in Asia. I don't know the exact location, but he's setting up the monkey, and the monkeys in these temples, they steal stuff from the visitors all the time. So here he is, he's like kind of like hanging his cell phone out. This monkey goes, grabs the cell phone, goes away, and not going too far away, gonna go and sit in this wall and wait for the guy to come and approach him. The second part of this video is even more amazing, where the guy is now trying to negotiate a deal with the monkey. He's offering the monkey first, I think, peanuts, and the monkey has clearly seen better options in his day, so like he's not going to be interested in peanuts. The guy, he starts to offer him bananas. We all know monkeys love bananas, but he's also probably eyeing some better reward. So he's keeping up on the bananas. Now he's offering him eggs, and I think that's what maybe he has settled for, is like he likes eggs or something. So he'll take the eggs, few things, good sensory motor coordination, catches it very well. Now the best part, he gives him kind of a look and leaves the phone intact. So this behavior to me is kind of really remarkable, because the monkey could have taken another approach. He could have thrashed, the phone or something, but he doesn't do that. Probably because they're already trained to, they know that if he did that, then next time the humans are going to have a very different approach in getting the phone back from the monkey. So in some way, the monkey has sort of, through training, through experience, many different ways, built a model of the agent, which is this human being, of how they might act and what to do to get reward out of it. And this is how I think I kind of see humans act all the time. Like I'm acting in a very specific way right now because I'm in a CVR seminar. I might not act in the same way when I go back home or if I'm in a pub or something like that. So like we adapt ourselves and we have a very different model of the environment that we live in, and we execute the model in some way in our head to make predictions of what might happen and what might be the best way to optimize on some dimension. So I think in a way, the kind of answers that I'm trying to get out of my research on high level visual cognition is like, how is that model executed in the brain? So I need a model, and I need to know, like, in terms of neurons and firing of neuron and interaction with different areas, how is that playing out in the brain? So as I was saying, also, like this involves behavior a lot in terms of like, what kind of behavior am I going to probe? What are those behavior? So for me, the main motivating factor is that those behaviors should be something not idiosyncratic to monkeys. They should be something that humans can also do. And humans probably do that very similar to monkeys. So that is a regime of behavior that I'm interested in. So I want to test humans in multiple behaviors, get human behavioral patterns on that, test monkeys on the same behavior, and find out this really nice spot where humans and monkeys are performing the same behavior in a very similar way. Because I think those are the more interesting paradigms where neural data from monkeys can benefit models of human cognition. So that's kind of the overarching theme that I'm trying to propose for my own research program, which is like, let's do these experiments, similar experiments in both humans and monkeys, and find out these common regimes. And also, if you think about it, like if I think about it, I find like maybe another factor to incorporate in this is that if those behaviors are clinically relevant, then it kind of adds more value to this whole program as well. So I think that's another factor that I want to include in the considerations when I start to design these experiments. And actually, I have developed a lot of pilot studies on this, and there's a lot of data available already to sort of go into asking what kind of models are there to start predicting those data. So as I initially set this up as like two different things that I mentioned, like object motion recognition, prediction, and visual reasoning. Like these are kind of like the sort of the compartmentalization of the visual cognition that I have in my head right now. So again, when I move from core object recognition to this sort of slightly more complex stimuli and behavior, the task complexity is obviously going to increase. With that, I might have different brain components that could support the task. So probably not ventral stream is not going to be enough. And also the models that will be explaining these tasks are going to be more and more complex. So these are the three things that I sort of expect when I move from core object recognition to this more complex tasks. Okay, so just the last 4 or 5 minutes, I think I have left in the talk. I'll just try to give you a sense of the kind of data that I've already collected in the last couple of years, the kind of data that will be part and parcel of the first sort of papers coming out of the lab and the first questions that the lab is going to try and answer. So one is sort of identifying these additional brain circuits that support dynamic vision. And the moment I even say the word dynamic vision, the things that comes to my mind first is that the object was stationary in the first case. Now the object is moving around. So, and I started, and I kind of like, this always kept coming to me when I was doing these experiments where I would, like, as I told you, like I can implant arrays in IT, and I can tell you like, okay, like you can decode out what object it is. It's a bear, and at some point in IT, you can tell that it's a bear. This is the kind of data that I was showing you before. It occurred to me like, okay, I'm never going to see a bear like this in real world, which is like stationary at one point for a long time. So the bears are moving around in the world. So like, what is IT doing when the bear is moving around? It was kind of like initially sort of a curiosity-driven question that I had. So I had this stimuli where the bear was sort of like moving around in the screen and not just like stationary. And to me, what was kind of interesting to find was that I can use the same population to decode the fact that the bear is moving to the right and not to the left. So that information is already contained in the same population. So that was kind of surprising to me, not too much because these are objects and motion of objects are very important property of an object. So the area that has representation of object identity might also have object motion. This is not a big surprise. The surprise that came to me was like, if I this is just to say that I have a lot of pilot data that needs modeling. So I'm super excited to collaborate with folks also at York to start modeling this data. But the thing that I was kind of interested in is that if I think of motion, the the terms that come to my mind is like dorsal stream, area MT, area V5, MST. Like this, I'm recording in IT. So what's happening? Like, are these interactions between dorsal and ventral stream important for this particular neural signature that I'm seeing? So this is one of the neural experiments that I want to pursue in the beginning, is that architecturally we kind of know where MT is situated. So maybe I can do some perturbation experiment in a very specific way in MT and ask what happens to this signal. And I have actually done a lot of recording in the dorsal stream in my PhD work. So I think that kind of makes me sort of suitable to do this kind of experiments where I can start, really perturbing these systems and asking like, what is happening to this signal in IT, which might be able to give us some sort of direction to this sort of ventral dorsal models that might be the next generation of models that are coming out. So as I said, like there's some control that is available because of these areas that are very visual, so we can make very controlled stimulus set, sort of tackle these kind of questions. And the second part was like this complex visual reasoning. And as I showed you this task of the monkey, you might be thinking that this is too complex of a task. Like, how can we actually do this kind of task in monkeys? So I've been developing some of these tasks in the lab. So for example, one task could be a likely event versus an unlikely event task. So you see a video like this, there's a ball that is moving, so the motion vectors are all there. And this seemed like a very likely event. Two balls came from both sides and just crossed. Whereas this is an unlikely event. And there is some level of just not following the motion parameters involved in this behavior. So you saw the white ball coming from the right side and kind of like moving away. That's not really obeying the physics of the ball if the monkey or the human has a good model of what the physics is. So these kind of tasks, I think, can be used to probe some of these cognitive, visual cognitive behaviors. This is one kind of task. Another task is like whatever has been used a long time for kind of tapping onto intelligence, quantifying intelligence. So a very caricaturized version of Raven's progressive matrix can be like, you have these three sequences, and I show you two options after showing these three images, like the top or the bottom, and no instruction, nothing. People will choose the top because it's 1, 2, 3, and then 4. You can do the same thing with objects. You can show apple, apple, apple. Choose this. But imagine if you change the rule from like all of a sudden, if you show cars here, then this might become the actual correct answer. So like monkeys can be trained to do this kind of task. And this is what I've been spending a lot of my time trying to do this. And I think these tasks can be sort of a gateway to sort of asking some of the more complex kind of reasoning questions that are there. And actually, this can be paired with the visual sort of motion task. So the same progressive matrix idea is implemented in this task where you can see a bunch of frames, one after the other, which is like this dog chasing the ball. And so I can stop the video right there at some point, and I can give you two options, like one where the dog has the ball in its mouth, and one where the dog is still chasing the ball. So like humans will have a preference based on what video I've shown to tell what's the most likely next frame or something. So like these tasks are very naturally occurring to the monkey as well. Like they behave quite kind of like spontaneously to these kind of their training of these tasks go very smoothly from my experience. So these are the kind of domains where I want to really expand the monkey behavioral testing. So as I said, this can be paired. I think York is a great, the whole strategy of the visual neuroscience building to sort of like have these arenas and the monkeys doing this kind of behavior is very, very attractive to me. And so I really am looking forward for the building to be built. If anyone has any access to the pedal, please press it so that the building gets built faster, if possible. I can pair these tasks with chemogenetic perturbation that can be executed in the cages, and also the wireless recording stuff that I've also developed and potentially can be added to the same setup. So last slide, as I said, I'm going to improve. I'm going to change this topic to like dynamic visual cognitive task, potentially recording from more brain areas. As I said, dorsal would be one part. And as we do that, the kind of models that are going to come up are going to be different. They're going to have additional component to it. And so I'm really looking forward to that. And here is a picture of my lab. And I have had a great time in the last 5-6 years working with amazing colleagues Some of them actually have moved on to Canada that I'm also super looking forward to working with them. So one of them is like Puya here, he's at McGill. So hopefully I'll have some more collaborations with him doing some of the control work that I showed you. Here's Jim, and I think I'm going to continue having collaborations with Jim and other members of the Ricardo Lab, but I'm also super looking forward to sort of develop these other ideas in my own ways also, and collaborating with many folks at York and specifically in CVR, because this is all vision. And I think I was just telling Richard that I'm a little bit scared actually. So many vision people, so I have to really, think about what I'm saying, because I'm afraid I'm going to say a wrong thing about vision. So it's going to be good for me in a lot of ways to be really sort of like surrounded by so many of you wonderful folks. So thank you all for listening, and I'm happy to take questions.